You know, it is completely wild to think about the fact that inside of you right now, as you're listening to this, there are millions of microscopic, literal suicide bags just waiting for a specific chemical signal to destroy themselves.
Yeah, I mean, it sounds like a tiny microscopic espionage thriller playing out in your bloodstream.
Right.
Or maybe some bizarre science fiction concept.
It really does.
But that level of ruthless efficiency is just standard operating procedure for human biology.
And that is exactly what we are unpacking today. Welcome to our deep dive. We are exploring the unit of life, the fundamental building blocks of you, me, and literally every single living thing on this planet.
Absolutely.
The mission for this deep dive is to strip away the dense academic jargon and look at this microscopic world for what it actually is. Not a static textbook diagram, but a bustling, highly efficient, and, sometimes brutally pragmatic hidden city.
And to see that city, we have to invite you, the listener, to zoom all the way in with us. We are leaving the macroscopic world behind entirely. Right, we're going to ignore the world of arms and legs, leaves and branches, and focus purely on the invisible engines that are working overtime this very second to keep you alive.
What I find so fascinating about our fundamental understanding of this invisible world is that it didn't start in some grand, well-funded laboratory.
No, not at all.
It started in the 17th century with a Dutch public official named Antony van Leeuwenhoek. He was essentially just a hobbyist. He just liked grinding glass lenses in his spare time.
He was a master of his craft, though. I mean, he ended up constructing hundreds of microscopes, but we really have to distinguish the technology he was using from what came slightly later.
Right.
Leeuwenhoek was using simple microscopes, and when we say simple, we mean it consisted of a single biconvex lens.
So mechanically speaking, he was essentially using a very, very powerful magnifying glass.
Exactly.
You'd hold this single piece of precisely ground glass right up to your eye and mount a specimen on a tiny needle-like screw on the other side.
And just by bending light through that one piece of curved glass, he could achieve a magnifying power of up to 200 times.
Wow.
Yeah, that opened up a previously invisible universe. But the real structural leap happened with the English scientist Robert Hooke. He developed the compound microscope.
The name compound sort of gives away the mechanism there.
Yeah, it does. Hooke used two lenses in tandem to compound the magnification, bending the light twice to pull the image even closer, which is, fundamentally the same architecture you see in a high school science lab today.
Although modern science eventually hit a physical limit with glass lenses and light, didn't it?
It did, yeah. A standard compound microscope might max out around 2,000 times magnification because light waves can only resolve so much detail.
Right, there's a hard cap.
Exactly. So to go deeper, modern electron microscopes ditch light altogether. They fire beams of electrons using powerful magnets to bend the beams instead of glass.
And that gets you what? Massive magnification?
Oh, yeah, achieving magnification of over 200,000 times.
That jump in scale is staggering. But going back to Hooke, he is working with his compound light microscope in 1665.
Right.
And he decides to examine a very thin, translucent slice of cork. And looking through those compound lenses, he sees these tiny, empty box-like compartments all piled up in a rigid grid.
He was looking at the microscopic honeycomb that made up the bark of the cork oak tree. And his observation gave us one of the most famous words in all of science.
Because looking at those sparse, empty little boxes, Hooke was instantly reminded of a monastery. Specifically, the small, bare, unadorned rooms that monks lived in.
The cells.
Right, those rooms were called cells. So he named the little cork boxes cells. But the great irony of this foundational discovery is that Hooke wasn't actually looking at living cells at all.
No, not at all. The cork was entirely dead. All the living substance that once filled those boxes had dried up and vanished.
So he was basically flying over a ghost town.
Exactly. Looking at the empty walls and abandoned foundations, seeing those dead structures was a monumental first step, but it didn't explain how a living creature actually functioned.
Which means we need a massive shift in scientific thought to figure out what those little boxes actually do. And that shift happens in the 1830s with two German scientists.
Right, Matthias Schleiden and Theodor Schwann.
Yeah, you have Schleiden, a botanist studying plants in 1838, and Schwann, a zoologist studying animals a year later in 1839.
They are looking at two vastly different kingdoms of life.
And this is where my mind gets slightly blown. I mean, a mango tree and a frog look entirely, completely different.
Obviously.
They behave differently. They survive differently. Yet Schleiden and Schwann realized that both the tree and the frog are running on the exact same underlying biological architecture.
It's incredible.
I used to think of it like building a tree and a frog out of the same biological Lego bricks, but that feels almost too static. It's more like they share a universal operating system.
Oh, I like that.
Right. The hardware, like the frog or the tree, looks different, but the fundamental underlying code operates on the exact same universal rules.
I like the operating system analogy much better. And they formalized those universal rules into what we now call the cell theory.
Okay, let's break that down.
Sure, so they established that all plants and animals are composed of cells. If you take a piece of a mango tree or a muscle fiber from a frog and put it under a microscope, you will always see that cellular structure.
Always.
Right. That leads to the first major pillar of the cell theory. The cell is the smallest structural unit of all living things.
But structure alone doesn't explain the movement of a frog or the growth of a tree.
No. Which brings us to the second pillar. The cell is the unit of function. Every single action an organism takes is actually just the cumulative work of its cells.
So when a frog jumps, it isn't magic.
Exactly. It is the physical contraction of millions of individual muscle cells pulling in unison. When that frog digests a fly, it is because specialized cells in its gut are physically manufacturing and secreting digestive enzymes.
And when the mango tree turns sunlight into sugar, that photosynthesis is happening strictly inside the cells of its leaves.
Spot on. The cell is doing the actual physical work of living.
So they aren't just the bricks of the building, they are the workers inside the building, constantly maintaining it.
That's a great way to put it.
But workers get tired. Workers die. And that brings us to the third rule of cell theory, added later in 1858 by Rudolf Virchow.
Yes, Virchow. He completely upended the idea that life just spontaneously appeared from nowhere.
Which was a popular idea back then.
It really was. But he stated that all cells arise purely from pre-existing cells.
It's the concept of continuous replacement. The body of that frog or that tree, or you, the listener for that matter, is composed of millions upon millions of cells that are constantly dying and being replaced.
And they are replaced by new ones formed solely by the division of younger cells.
It is an unbroken chain of life. Every single time you heal from a paper cut, that is Virchow's rule in action. Pre-existing skin cells are dividing to bridge the gap.
And if you trace that chain of division all the way back, practically all complex life starts as a single solitary cell.
Like the frog started as a single fertilized egg, a zygote.
Exactly. The towering mango tree started as a seed with an embryo, which also originated from a single fertilized cell. One cell divides into two, two divide into four, and eventually you have a fully formed, incredibly complex organism.
But if these tiny cells are responsible for every single function in a tree or a human, they have to be incredibly active.
Oh, they are doing a massive amount of heavy lifting. And to maintain that extreme level of metabolic activity, they are bound by very strict physical limitations.
Limitations that explain why they are so bizarrely tiny.
Yes, the scale is almost hard to comprehend. We are talking about bacteria that can be as small as 0.3 micrometers. Human red blood cells are around 7 micrometers.
There are a few wild outliers, of course.
Right, like a nerve cell can extend from your fingertip all the way up your arm to your spinal cord. Or the largest single cell in the world today, an unfertilized ostrich egg.
Wait, just this central yellow yolk sphere is one single massive cell.
It is, but those outliers survive because of very specific environmental conditions, or, in the case of the egg, a lack of active metabolism until development begins.
Okay, that makes sense.
For the vast majority of active cells, they must remain microscopic for a single reason, and that is efficiency. It all comes down to the geometry of the surface area to volume ratio.
Let's actually walk through the math on this, because understanding this ratio is the absolute key to understanding why you aren't just one giant human-sized cell.
Yeah, let's do the cube example.
Imagine you have a cube, and every side of this cube is exactly 2 millimeters long.
Okay, tracking.
If you calculate the total surface area, like the outer skin of that cube, you get 24 square millimeters.
Right.
Now, if you take a knife and slice that exact same cube up into 8 smaller 1 millimeter cubes, the total amount of stuff inside, the volume, stays exactly the same.
But because you've just exposed all those new inner sides, the total surface area instantly doubles to 48 square millimeters.
You've maximized the borders without changing the internal mass.
And for a cell, survival is entirely dependent on its borders. A cell needs to communicate with its environment. It requires rapid, constant diffusion of vital substances.
Nutrients and oxygen have to cross the border and get to the center of the cell quickly.
And just as importantly, metabolic wastes and carbon dioxide need to evacuate quickly.
Imagine a massive city where the central downtown area is 10 miles from the nearest port.
That's a great analogy. If supplies arrive at the port, it takes forever for them to travel inward to downtown. And it takes forever for downtown garbage to travel outward.
Exactly. If a cell gets too large, its internal volume becomes a massive downtown area, but its outer surface area, the port, isn't big enough to handle the traffic.
The center of the cell would essentially starve for oxygen or be poisoned by its own trapped waste.
So by staying microscopic, the cell ensures that no internal part is ever too far from the outer border.
Plus, if a tiny cell gets damaged, the problem is localized. You just replace that one tiny cube rather than losing the entire structure.
It is an incredibly resilient design. But because cells rely so heavily on exchanging materials across these borders, the actual architecture of the border has to adapt based on the environment the cell lives in.
Which introduces a massive split in how biologists classify life. And the first major distinction is between prokaryotic and eukaryotic cells.
And the defining feature here is how a cell protects its most valuable asset, right? Its genetic code.
Yes. A prokaryotic cell is structurally primitive. It operates like an open floor plan.
Just wide open.
Yeah, the genetic material, a single length of DNA, is just floating freely in the central region of the cell. It has no nuclear membrane wrapping it up, no dedicated secure vault. Bacteria are the classic example of prokaryotes.
The DNA is just out there in the open, vulnerable but very accessible for rapid reproduction.
Contrast that with eukaryotic cells. These are cells with a perfectly formed nucleus.
Right.
The DNA is securely enclosed inside a double nuclear membrane. It is a highly protected vault. Plants, animals, humans, we are entirely made of complex eukaryotic cells.
But obviously, a human being is fundamentally different from a leafy green fern.
I should hope so.
So, if we both use eukaryotic cells with protected DNA vaults, what is structurally different about our cells? Like, if I'm building a plant cell versus an animal cell, how do I change the borders?
Let's start with the animal cell. Its outermost boundary is the cell membrane, sometimes called the plasma membrane.
Okay.
It is very thin, highly flexible, and incredibly alive. It is semi-permeable, which means it acts like a selective border patrol, actively regulating exactly which molecules are allowed to enter and leave the cell.
And that flexibility is why animals can have soft tissue and bendable joints. But a mango tree doesn't have a skeleton to hold it upright against gravity. How do its cells compensate for that structurally?
Well, plant cells have that same living cell membrane, but surrounding it on the absolute outside is an additional layer, the cell wall.
Right, the cell wall.
And this wall is made of cellulose. It is entirely non-living. It gives the plant cell a rigid, definite shape, acting almost like an external skeleton for every single cell.
Interestingly, the cell wall is freely permeable, isn't it?
Yeah, it doesn't filter anything. It lets water and solutions pass right through. The living cell membrane underneath still handles the actual selective filtering.
So plants construct an extra suit of non-living armor on the outside. What changes on the inside to support that heavy armor?
Inside, plant cells feature massive vacuoles. These are clear, membrane-bound spaces filled with a liquid called cell sap, which is mostly water with various dissolved substances.
And these serve a structural purpose.
A critical one, turgor pressure. They fill up with water and press outward against that rigid, non-living cell wall, keeping the cell incredibly firm.
Oh, so that internal water pressure is literally what holds the stem of a plant upright.
Exactly. If you forget to water a houseplant and it wilts, it's because those microscopic vacuoles have drained and lost their turgor pressure. Animal cells, on the other hand, rarely have vacuoles, and if they do, they are tiny and temporary.
But animal cells have a different specialized tool that plant cells completely lack: the centrosome.
Right. The centrosome contains two small structures called centrioles. When an animal cell prepares to divide, those centrioles migrate to opposite ends of the cell and form spindle fibers.
Imagine microscopic bungee cords that reach in, grab the chromosomes, and pull them apart so the cell can split evenly.
Yeah, and plant cells somehow manage to coordinate their division without using centrosomes at all.
And there is one more structural marvel uniquely found in plant cells, right? Plastids.
Yes, specifically green chloroplasts.
I want to highlight something really specific about how we talk about chloroplasts here, because it's easy to misunderstand their function. People often call them the kitchen of the plant cell. But a kitchen is a terrible analogy for what is actually happening.
It really is. Think about the mechanics of a kitchen. You take ingredients that already exist, like flour, eggs, sugar that some plant or animal already produced, and you apply heat to modify them into a cake.
You are just rearranging existing energy.
Right. The chloroplast is doing something far more magical. Chloroplasts contain chlorophyll, which physically traps raw solar radiation. It takes that kinetic energy from the sun, grabs carbon dioxide out of the air, pulls water up from the roots, and literally manufactures food, starch, and sugar from scratch.
So it isn't a kitchen modifying food. It is an organic manufacturing plant spinning highly energetic sugar out of thin air and sunlight.
It is the ultimate self-sustaining engine. But whether you are looking at a rigid plant cell with its solar factories or a flexible animal cell with its centrosomes, everything inside that outer membrane is suspended in a highly active fluid medium.
Yes, biologists call this the protoplasm. It is the total living substance of the cell, encompassing both the cytoplasm and the nucleus.
And the way this living substance is studied is almost philosophical.
Because protoplasm is constantly reacting, constantly moving. It is the physical manifestation of life. If biologists try to pull it out and do an accurate chemical analysis on it, they fail.
Yeah, the very moment you remove it from the living organism or apply harsh chemicals to study it, it ceases to be protoplasm. The act of dismantling the machinery stops the living process entirely.
It reminds me of trying to understand how a beautiful, complex mechanical watch ticks by smashing it with a hammer.
Ooh, yes.
You can analyze the resulting pile of gears under a microscope all day long. You can categorize the metal, but you just have a pile of dead gears. You no longer have the ticking. You've lost the animating force.
That captures it perfectly. Protoplasm isn't just a static soup, it is the ticking watch. And embedded within the cytoplasm, which is the portion of the protoplasm outside the nucleus, are all the specific cell organelles.
These little organs perform the highly specialized labor of the cell. Let's look at a few of these little organs, starting with the power plant, the mitochondria.
Mitochondria are minute, double-walled bags responsible for cellular respiration. And here, that brilliant mathematical principle of surface area makes a spectacular return.
Oh, I love this part.
The outer wall of the mitochondrion is smooth, but the inner wall is thrown into multiple, complex, inward-projecting folds called cristae.
So instead of a smooth inner balloon, it's crumpled and folded in on itself.
Huh.
By doing that, the cell tacks vastly more surface area into the exact same microscopic space.
And that extra surface area is critical because that inner membrane is where the chemical reactions happen that release energy in the form of a compound called ATP.
So the more surface area those cristae provide, the more ATP the mitochondrion can crank out.
Exactly. The heat your own body is generating right now, keeping you at 98.6 degrees, is the direct physical byproduct of millions of these little folded membranes burning fuel.
So we had the power grid online. But a bustling microscopic city also generates threats. Which brings us back to the hook from the very beginning of this deep dive, the suicide bags, the lysosomes.
Lysosomes are small, membranous vesicles drifting through the cytoplasm, and they are packed with up to 40 different types of extremely powerful digestive enzymes.
Their daily job is intracellular digestion, right?
Yeah. If a foreign substance, like a harmful germ, breaches the cell membrane, the lysosome zeroes in, engulfs it, and unleashes those enzymes to chemically dissolve the threat.
They are the immune system's microscopic infantry. But why do we call them suicide bags?
Because of their intensely pragmatic, darker function. If the cell itself becomes too old or sustains severe injury, or if the entire organism is starving and desperate for raw nutrients, the cell issues a command.
And what happens?
The lysosomes rapidly burst open all at once. They flood their own cell with those 40 destructive enzymes, dissolving the entire cell from the inside out. They sacrifice the individual cell to prevent the spread of damage or to provide emergency fuel for the greater organism.
It is a brutal, selfless efficiency. We have the power from the mitochondria, the defense and recycling from the lysosomes. But none of this works without a command center coordinating the chaos.
Right, and that is the nucleus. The nucleus regulates all the life processes we just discussed. But its most profound responsibility is protecting the blueprints for life itself.
Inside the nucleus, you have a dense fluid called nucleoplasm. Suspended within that fluid are chromatin fibers. Now, when the cell is just going about its daily business in a resting state, these chromatin fibers just look like a messy, tangled net of DNA threads floating in the fluid.
But Virchow's rule demands that cells must eventually divide, and when it is time to replicate, you can't just hack a tangled net in half and hope both new cells get the right information.
The process has to be flawless.
Exactly. So that messy net dynamically transforms. The threads rapidly condense. They meticulously spool themselves up into distinct, thick, ribbon-like structures called chromosomes.
And these chromosomes carry the genes. They are the physical bearers of heredity, dictating every characteristic of the species.
Every species has a highly specific fixed number of these chromosomes. Humans, for instance, have exactly 46 chromosomes, arranged in 23 pairs.
When a human cell divides, that exact set of 46 blueprints is perfectly duplicated and passed on.
Which ensures that the newly formed cells know exactly how to be human cells, how to build mitochondria, how to formulate lysosome enzymes, and how to maintain the living protoplasm.
It is the ultimate relay race. From that very first single zygote we discussed earlier, all the way down to the trillions of cells coordinating inside you today, the information transfer has remained unbroken.
So when we step back from the microscope, what do we actually see? We started with Robert Hooke looking at dead, empty wooden boxes, but now we see the cell as a dynamic, autonomous city.
We see mitochondria frantically cranking out ATP by folding their inner walls to maximize surface area.
We see lysosomes patrolling the borders, ready to dissolve themselves to protect the tissue.
We see the nucleus neatly packaging its chaotic chromatin net into 46 flawless chromosomes to pass on your genetic code to the next generation of cells.
And for you, the listener, the ultimate takeaway is a profound sense of scale and constant invisible motion. Every single time you move your arm, every time you take a breath, every time you simply process a thought, it is entirely the result of this cellular machinery.
You are not a single, solid, static object. You are a highly coordinated walking colony of approximately 37.2 trillion individual living cells.
37.2 trillion separate entities acting in perfect unison to create the illusion of a single being.
It is mind-bending. But I want to leave you with one final thought pulled straight from the realities of this microscopic world.
Sure, lay it on us.
We talked heavily about how cells die and are replaced. Our sources note a very specific timeline for human red blood cells. The cells furiously ferrying oxygen through your veins right now. They only live for about 120 days.
Wow.
Yeah, after 4 months, they die and are entirely replaced by newly manufactured ones.
It is a shockingly rapid turnover rate for something so vital.
Exactly. So if Virchow's rule holds true, if the formation of cells from pre-existing ones is a never-ending chain, and millions of your cells from your blood to your skin to the lining of your stomach are bursting, dividing, and being replaced this very second, well, how much of you is actually the exact same physical material you were a year ago?
That's a trippy thought.
When we look in the mirror, we expect to see a solid, unchanging framework. But underneath that visible surface, you are a river of constant microscopic turnover. Are you looking at a static photograph of yourself, or are you looking at a 37 trillion cell city that is quietly, relentlessly rebuilding itself from the inside out?